only time I have like not zoomed. Oh, okay. Well, I'm sure whoever was on Zoom got off by now. Am I sharing the screen? Okay. I don't know what I'm doing here. All right. It should be, I'm screen sharing. Okay. All right. Sorry, y'all. All right. Ooh. Oh, it's playing. What is it doing? It's a bad technology day today. All right. I'm going to try not to blast y'all out.
The citrate cycle is the metabolic engine of the cell. When energy in the cell is low, the citrate cycle is critical to the production of energy from fatty acids, proteins, and carbohydrates. The fuel for the citrate cycle is acetyl-CoA. The exhaust is carbon dioxide, and the work performed is the transfer of electrons. In each turn of the citrate cycle, 1 acetyl-CoA is oxidized, and 8 electrons are transferred to 3 NADH and 1 FADH2. In the process, 1 GPP is also produced by direct phosphorylation of GDP. The NADH and FADH2 are used to power the electron transport system, resulting in the production of approximately 10 ATP for each acetyl-CoA that enters the citrate cycle. The flow of acetyl-CoA into the citrate cycle is controlled by the pyruvate dehydrogenase enzyme complex. The pyruvate dehydrogenase complex has a molecular mass of almost 8,000 kilodaltons and contains nearly 80 individual protein subunits. Pyruvate dehydrogenase uses pyruvate from the glycolysis reactions and metabolizes it to acetyl-CoA, fueling the citrate cycle. The pyruvate dehydrogenase complex consists of 3 interdependent enzyme subunits that use pyruvate, CoA, and NAD+ as substrates, and in a 5-step reaction mechanism, generates carbon dioxide, acetyl-CoA, and NADH. 5 different coenzymes are needed to enable the enzymatic activity of the pyruvate dehydrogenase complex. Nicotinamide adenine dinucleotide, derived from vitamin B3, flavin adenine dinucleotide, derived from vitamin B2, coenzyme A, derived from vitamin B5, thiamine pyrophosphate, derived from vitamin B1, and lipoamide. The product of the pyruvate dehydrogenase reaction, acetyl-CoA, is used to fuel the citrate cycle. The first reaction in the citrate cycle commits to the acetate unit of acetyl.
Okay, I want to pause it for just a second. So when do I want to convert pyruvate to acetyl-CoA? When I have a low energy in the cell. So I want you to kind of be thinking about regulation before we even talk details. What do you think might inhibit this reaction from happening? High levels of ATP. So high levels of ATP will inhibit pyruvate dehydrogenase. Then you build up pyruvate. So what's going to happen? You're going to end up shuttling those metabolites into different pathways, right, to be used for different things. Okay, how do I, oh goodness.
Acetyl-CoA to oxidative decarboxylation by catalyzing its addition to oxaloacetate, forming citrate. Oxaloacetate is regenerated in the last step of the cycle, so it can be reused as the substrate for the first step. Note that the metabolic cycle is referred to here as the citrate cycle because citrate exists predominantly in the deprotonated form at physiological pH and is therefore not an acid in living cells. Other names for the citrate cycle are the tricarboxylic acid cycle, or simply TCA cycle, and the Krebs cycle, named after the biochemist who first described it, Hans Krebs. The citrate cycle contains 4 coupled redox reactions. They use the oxidized forms of the coenzymes NAD+ and FAD as electron carriers, reducing them to NADH and FADH2. NADH and FADH2 are then oxidized by the electron transport system, producing ATP. Citric acid is used commercially for a wide variety of purposes. be isolated in great quantities from the fungal mold Aspergillus niger using fermentation methods that exploit the citrate cycle reactions. High levels of citrate synthase are found naturally in the fungus. When the other citrate cycle reactions are inhibited under certain culture conditions, citrate is overproduced by the fungus and secreted into the culture medium.
So you can hijack different living systems in order to do what you want them to do, right? We can make bacteria make insulin for us. We can make mold make citric acid, right? Part of biochemistry is using that to our evolutionary advantage, right? Okay, so, oh. Okay, so remember in the video, they said that you had electron carriers, right? So those electron carriers are a little bit unique. Remind me real quick, if we're talking oxidation reduction, what is oxidation reduction? Right? Is loss, well, loss of electrons, what else? Loss of hydrogen, gain of oxygen. Reduction is gain of electrons, loss of oxygen, gain hydrogen, right? Okay, so what happens in our molecules? We have NAD plus, right? NAD plus is going to pick up electrons from our substrates, right? So they're actually going to give up hydrogen atoms. So most of the time in all of these oxidation reduction reactions, you're going to hear the words dehydrogenase, right? And so what does that tell you is going to happen? You're going to lose some hydrogens, and who's going to pick up the hydrogens? These guys, right? So if we have NAD plus picks up some of those electrons in the form of hydrogen ions, and some of them as a hydride ion. Have y'all seen a hydride ion? It's carrying 2 electrons and a negative charge. Yes. Ooh, some head nodding. I like it. It's the chemist. It's the chemist. So then what happens? What do you make? NADH. Whoa. Getting caught on my folder here. NADH. And what else? That's too many hydrogens, right? Balance that sucker. All right, so what was my other one? FAD, right? So FAD is going to pick up basically 1 electron, and it's going to pick up a hydrogen ion. So then what are you going to get? DH. You're going to pick up, it's actually a 2-step mechanism. I should have written that. It's actually a 2-step mechanism. So we'll just put, well, you don't need to know the 2 steps, just do that. What do you get? FADH2, okay? So now you have those energy carrying molecules. So what makes one molecule pick up electrons and others drop off electrons? What's up with that? What is it? Close. What? Very close. So it has to do with our structure. So it's all about the potential of those molecules and how much they want those electrons, right? So it's a little bit about electronegativity. We call that our reduction potential. Your book has a table of the reduction potentials for all of the different substances that are in these pathways. And so if you look at the reduction potential, sometimes they not prime, right? Reduction potentials. Have y'all seen that before in any chemistries? No. Yes? No? No. All right. So if you want to know, are electrons going to be transferred? what you have to look at is the reduction potential of both your substrate, like whatever's carrying the electrons, and whatever you think is going to pick up the electrons. And you're going to subtract the two, and you're going to figure out, are those electrons going to move, right? So you do your acceptor, electron acceptor, minus your, wow, minus your donor. It's like the pen's not picking things up today, right? E naught prime. E, electron acceptor. See, it's like the pens. I gotta go slow. If I write fast today, I don't know. It's not liking me. And this is our donor. You could say E minus donor. Okay. And it's also related to free energy. Remember, we've talked like extensively about free energy. So there's a formula that relates the two. So our free energy is equal to, okay, so what is N? In formulas generally, what is N? I'm pointing to it with my finger. Number of something, right? So what are we doing in this example, in these problems? What are we transferring? Number of electrons. What is F? Faraday's constant. Which is, anybody know? What? 98 kilojoules per volt mole. Right. So when you have a difference in reduction potential in a redox reaction, you can transfer your electrons. If you don't have a difference in electron potential, it's not going to happen. So when we're done with all of this, when we get to the very end and we make ATP, you have to think about what has had to happen. We have had to move these electrons along all these different molecules that we're going to eventually learn until we get to our final electron acceptor. Anybody know what our final acceptor is? So our final. This is why it has to be in the mitochondria, is O2. Because if you don't have oxygen as a final electron acceptor, you are not going to be able to continue to pass those electrons along. If you stop having oxygen, you shut down the electron transport chain, everything backs up, citrate backs up, everything backs up. So eventually what's going to happen? O2 is going to get converted to, when it accepts electrons, and you got hydrogen ions floating around, you're going to eventually turn it into water, right? Which is something our bodies can handle, which is really nice. Okay, so that's kind of like the end of the story before we even talk about the whole story, right? All right. I can move. I can go. What? Say it again?
So she needs to go to the chiropractor with that backtrack.
The desk is good. The desk is good. As long as you have something to pull against, right? Okay, so we finished with glycolysis. We ended with pyruvate. But we know to enter into citrate cycle, what do we need? Acetyl-CoA. So we have to go through pyruvate dehydrogenase. Is pyruvate dehydrogenase part of glycolysis? Is it part of the citrate cycle? No. 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 So in order for pyruvate dehydrogenase to work, right, it's that huge big old complex. We're going to need 5 different coenzymes. What did you hear in those videos that these coenzymes are? A lot of them, not all of them. They're vitamins. They're vitamins, right? And you're going to notice that a lot in the different pathways that we talk about. The coenzymes that tend to be really important for those reactions are the vitamins that you'll see, you know, if you buy a bottle of vitamins, you're gonna see them. So if we're going to be using pyruvate dehydrogenase, where are we? No, we went through glycolysis, and we have pyruvate in the cytosol. The pyruvate is transported into the mitochondria, right? So we're in the matrix. This is the inner mitochondrial membrane. Inner, and this is the outer membrane, right? You biologists in here, right? Y'all got this? So then where are we here? We're in the matrix, right? Because the mitochondria is a double membrane organelle. So now we have pyruvate in the mitochondria. So now that we're in the mitochondria, now we can convert pyruvate to acetyl-CoA. Now that acetyl-CoA can go into the citrate cycle, right? And go through all those different reactions. So we're in the mitochondrial matrix. Mitochondrial. Thank you. Sometimes I go off on a tangent. M-A-T-R. In the matrix. I forget like what question I asked y'all. All right, so it's a complex in the matrix and we require 5 different coenzymes. All right, so remember we talked about our NAD plus and our FAD being redox reactions, right? And so these, you're gonna see a lot in different reactions that are in metabolism in the body. The citrate cycle and pyruvate dehydrogenase are not the only ones that use redox reactions, but it's a good example of it, right? So whenever we talk about our NAD plus, you're only showing a small portion of the molecule. Do you see this R, right? There's a whole big portion of the molecule, but we don't really care about that because that's not what's happening. That's not what's changing, right? We basically leave R as whatever doesn't change. And then we look at the rest of the molecule that's important. So what happens when we convert from NAD plus to NADH is this carbon is going to pick up that hydrogen and then you release your free hydrogen, right? Same thing in FAD. FAD is sequential. Remember how I said it was a sequential stepwise process. You're going to attach hydrogens on the nitrogens in the rings. It's not, you don't have to memorize structures. You don't have to memorize structures. Okay, so coenzyme A. Coenzyme A is critical in generating acetyl-CoA. So this is one of our B vitamins. So if you know of anybody who's older, like when my grandmother was still around, she used to go and get B vitamin shots. Anybody know old people who go and do that? So why? Why do they do that? They know? Makes them feel like amazing for like a week and a half. It's great. It's for energy. It's for energy production, right? So this is going to enhance our ability to enter into the citrate cycle to make ATP and electron transport chain and all that kind of stuff. And so really it helps the most with cells that are that you need to regenerate a lot of, like red blood cells. So it'll pick up their red blood cell count as well, which is really good. All right, so if we want to convert, into acetyl-CoA. So here's our coenzyme A. So what's going to happen in converting? What we need to do is to attach our acetyl to our acetyl-CoA enzyme, right? So what did we add on to the very end? An acetyl group, right? We have 1, 2, 3 carbons, not 3 carbons, 2 carbons, if I could count today, that we're adding on to our group here, okay? All right, so then we have thymine pyrophosphate. So what's a pyrophosphate? Why do we call it pyrophosphate? If we have pyrophosphate by itself, we call it PPI. Y'all seen that before? Because a phosphate group is just 1 phosphate, but pyrophosphate is 2 phosphates. So if you look at the structure of thymine pyrophosphate, we'll have 2 phosphates, right? So this is another one that's required for our pyruvate dehydrogenase. And this is another B vitamin. But you notice commonalities, right? More B vitamins. Alpha lipoic acid. So this is going to give us that disulfide that's going to participate in redox reactions. There are lots of disulfides that interact in order to make oxidation reduction happen, right? Remember when we talked about proteins and we said you could get disulfide bonds that occur in between beta strands or things like that in order to lock things into a particular orientation? Did we talked about curly hair and like straight hair, didn't we? Yeah. And what are you doing? You're just breaking those bonds chemically and then putting a curling iron or a curler or whatever it is, and then chemically reforming them. And now the strands can't slide past one another and the hair stays in a particular way, right? It's the same thing. So this is just giving us that reducing potential in order to go from our disulfide bond to our reduced form here, so that we can make our acetyl-CoA and send that acetyl-CoA to the citrate cycle. So all the reactions that occur in the pyruvate dehydrogenase complex are all oxidation reduction reactions. You're going to see that over and over and over again. So this was that super complex model that you saw, right? So we said that there are 3 basic complexes to pyruvate dehydrogenase. And this is going to be highly, highly regulated. So look at the different substrates that are required to make this happen. Look at NADH. If we require NAD+ as a cofactor in order to make pyruvate dehydrogenase happen, what happens if we hit high levels of NADH? That means that we're going through and we're making tons of acetyl-CoA, but do we need it? No, it's like a feedback mechanism that's going to tell us, okay, we've got tons of NADH, so why are we going through the citrate cycle, right? The same thing at ATP levels, the same thing about acetyl-CoA levels. These are all substrates that are going to tell us basically that we've backed up the system, that we have too much being made and we need to slow down, right? And then so what's the body going to do with all of our substrates? If we don't go through citrate cycle and we don't go through electron transport chain, where are they going? More than likely. What's that? Like what? Well, what feeds into the citrate cycle? Fats and amino acids, right? So then you're going to end up shuttling it out to go and make those things, right? So whatever feeds into it, you have to think you can also shift the flux to those things because they were double-sided arrows. Yeah? Okay. All right. The other thing is that it can be regulated by phosphorylation. So what are these? Phosphorylation is a different mechanism for regulation than the others. Why? Remember we said there are different ways that you can regulate enzymes? What are the different ways? Post-translational modifications. And you can also have allosteric control, which are the other ones. So is this an overall favorable reaction for the cell? Yes. How did we make it favorable? We coupled reactions. If you couple reactions, you can drive them forwards. So those same concepts that we learned in, I don't even know what chapter that was. I feel like I should probably know that. They don't go away. All right. So there are different drugs that are out there that can inhibit reversibly and irreversibly all of these processes. Arsenic is one of them. So remember how we said we needed these disulfide bond right here? If you give someone arsenic, what's going to happen is that arsenic is going to bind to these sulfurs. So normally where you're supposed to be able to regenerate the disulfide bond and then use it in your pyruvate dehydrogenase complex. Here, when arsenic comes in and it binds to these sulfurs, it's locked in this position and it's not regenerated. So the lipoamide is not regenerated. So what happens to somebody who has arsenic poisoning? What's that? Yeah, you shut down ATP production. They get very, very, very sick. It actually tends to leach into water wells, drinking wells in third world countries and things like that. And so water testing is super, super, super important. And we're going to talk about, as we go along, lots of different inhibitors of not only the citrate cycle, but also the electron transport chain. And you'll recognize some of them. Okay. Y'all ready to tackle the citrate cycle? I got this. It's not that bad. It's not that bad.
I would say that. It's always that bad.
All right. So technically, by the time we get to the citrate cycle, we are in the presence of oxygen. So it is an aerobic pathway. And it's cyclic, and you only have 8 reactions. Look, it's even less than glycolysis, right? It's true. It's true. It's true. So what is the first product of the citrate cycle? Citrate. So what enzyme is going to catalyze the first reaction? Citrate. What's a synthase enzyme? It's going to make something, right? It's going to make basically whatever the name is before it. Whoo! So tell me about this reaction. What do we have for a delta G value? Negative 31.4. So this is a highly favorable reaction. What do you notice about this table that you didn't see in glycolysis? There's not like another column, right? It is super difficult to get into the mitochondrial matrix and assay parameters of metabolic reactions without messing them up. So we're not very good at it. And so we basically only have these delta G values. Oh, standard conditions. And the other one is like current conditions, like whatever the cellular conditions are. Standard conditions tend to be cellular, but not necessarily, right? Especially in terms of substrate concentrations. That's like the really big difference in regular standard delta G values and actual delta G values. Yeah. Okay. So then when we go, our next one, we're just going to do an isomerization where we go from citrate to isocitrate. We're not making anything too fancy. The next one, once we've made isocitrate, now we're going to do an oxidation reduction reaction. How do I know I'm doing an oxidation reduction reaction? I can look at this and find two ways that I know that. That's exactly right. We convert NAD plus to NADH, and that enzyme that performs that function is a dehydrogenase, right? And what is it taking the hydrogen from? Isocitrate. Okay. All right. So the next one, guess what kind of a reaction this is? Another oxidation reduction reaction. This one is alpha ketoglutarate dehydrogenase. So we have alpha ketoglutarate as our substrate, and we are going to convert NAD plus to NADH. We also are going to convert our coenzyme A. We're going to make carbon dioxide out of it. So this is part of why when we undergo cellular respiration, what do we exhale? Carbon dioxide, right? That's one of our waste products. Okay, so now we have succinyl-CoA. So succinyl-CoA synthetase. Synthetase is one where you are actually involved with a triphosphate, an ATP, a GTP, something like that, right? Because you can have a synthase and you can have a synthetase. They're very similar though. They're very, very similar. So we're going to make our GTP here, and we're going to have succinate as our product. So look at the next reaction. What are we doing? Succinate dehydrogenase. So then I know I'm going to have an energy carrying molecule. In this case, it's FAD going to FADH2. So what do you notice about this dehydrogenase reactions? Each one of them, we're making an energy carrying molecule, right? Okay, so we had succinate dehydrogenase, we make fumarate, we convert fumarate to malate. Then once we have malate, we convert malate to oxaloacetate. Where does oxaloacetate go? Back to the beginning. That's why it's a cyclic pathway, because this end product is also an initial substrate. So it gets used up very, very, very quickly. So that's why it will still happen even though we have a delta G value of, goodness, positive 29.3, right? That's not actual. Okay. All right, so our citrate cycle. Every time you'll see the citrate cycle drawn, you're going to see pyruvate dehydrogenase, but pyruvate dehydrogenase is not technically part of the citrate cycle, okay? But in order to start the citrate cycle, you have to convert pyruvate to acetyl-CoA. So when you're counting up how many, if I ask you something like how many NADHs are made, how many whatever molecules, energy carrying molecules are made in the citrate cycle, you don't count their NADH. But if we're eventually going to do this, where we start with one molecule of glucose and we track all of the energy that comes from one molecule of glucose if we start at glycolysis. So we would count everything in glycolysis, we would count the pyruvate dehydrogenase reaction, then we count the citrate cycle reactions.
Although, like we're talking about like pyruvate, are we going to go into the details of like how the amino acids are broken down to create acetyl-CoA?
You have to take biochem too.
Okay. I didn't know if we were going over it in this one.
No, we talk about amino acid metabolism in biochem too. Please take it. It's fun. Oh, I never posted. I need to post those contact stuff for y'all. Okay. All right. So what you should kind of do when you're looking at these reactions is kind of keep track of your carbons and keep track of what happens to those carbons as we go around in our pathway. But like, I don't want to start the reaction and then we're going to get cut short. So I think what we're going to do is we're going to stop here and we're going to start all of our reactions one at a time on Friday. Yeah? Wait, what?
I don't think we would ever get on a class together.
I know, but I don't want to go too fast. Yeah. I don't want to stop in the middle of the citrate. I'm not going to be able to finish the whole citrate cycle. I'm not.
Do you think we're going to get to chapter 11?
I don't know.
I don't know either.
I don't know. I hope we do, but I don't know. I don't know.
I was going to go ahead and start like, I don't know, like between, you know.
Yeah.
I don't want to do it if it's not going to be
Yeah, I don't know. Do your lab stuff. Do your lab stuff. Do other stuff
I did
to
last
prep.
time. I'm done everything.
You've done everything. Go back and study glycolysis. Go do the practice discussion questions at the end of chapter 9.
No, girlie. I don't want. I'm not trying to get something to do. I'm trying to knock everything out of the way.
All right.
Like I want the homework out of my. I don't want it in my sight. I don't want it in there. But I don't.